Briefly to start, I wanted to mention that we have been making some progress with the research outcomes of this program, and I hope that some of you that are participating in the program are continuing to make your projects ready for different poster presentations or even publications. And as an example, I'll start with sharing just a few details about a poster that we presented at the recent Louisiana Biomedical Research Network Bioinformatics annual conference, because it actually shares some similarities with the topic of today's webinar. So in this poster, we focused on a new data set that was collected and sequenced by the Noguchi Memorial Institute for Medical Research, Anita Gansa's group, and that was done primarily by Benedicta Mensa. But the focus of the poster was on the interaction of the apical membrane antigen rupturing neck protein 2 binding, which are candidates for vaccines. And so what's important about the efficacy of these vaccines is the genetic diversity of this particular of the antigen, which is the AMA1 protein. So one of the things that we wanted to investigate in this project was the mutational signatures that actually define this diversity. And one of the ways to define this diversity is to take a look at the variants in the perspective of haplotypes. And so I think this is a good connection to the project that we'll be talking about today, because it also speaks about haplotypes, but a little bit in a different context. So again, what is Plasmodium falciparum? For all of you that are probably familiar, it's a parasite actually, so that invades red blood cells, and that causes the clinical manifestation of the disease called malaria. And in the context of real prevention, many different approaches have been taken, including anti-malaria drugs that cause eventually the emergence of resistance. There are preventive measures like mosquito nets and eradication of mosquitoes and things like that. And so that reduces the transmission. And then one of the major additional focuses now is the vaccine approach. And so the vaccine approach has not been as successful as a lot of people have hoped for in the past. And that is primarily, again, linked to this challenge of diversity. And the genetic diversity allows the parasite, any kind of an infection pathogen, to evade detection of the immune system, which has been trained on a particular type of an antigen. And the idea of kind of suppressing, so increased immune pressure on that parasite or on that pathogen is a pressure that results in evolution, kind of selection of a particular type, of a particular strain. So what we did in this project, and I think this is going to be evident how this is similar to the project that we'll be talking about today, is take assembled sequences that already have been assembled from the reference, from mapping of those sequencing reads onto the reference genomes, and then from looking at all of the possible variations, creating these haplotypes. And so the haplotypes in this case refer to assembled sequences that contain multiple mutational signatures, which means that they contain specific mutations that occur together, right? And that was one of the things that was done to characterize population structure, because one of the hypotheses that were pursued in this project was whether this genetic structure actually defines kind of the prevalence of disease or this infection. And so as a result, we looked at independently occurring mutations, and then we looked at haplotypes which were connected into a network, and we tried to ask the question, well, could we find haplotypes that are unique or more prevalent in a population? And as a result, we've identified some interesting trends. So those mutational signatures could be affecting in a specific pattern. They could be affecting T cell and B cell receptors, and especially this could have an impact on the binding between these two proteins. But I think that this experience kind of validates this approach of taking mutations as kind of the result of immune pressure. So again, B cell and T cell receptors monitoring how they could be potentially affected by mutations is allowing us to look at mutational pressures. And then thinking about haplotypes, we're not just thinking about kind of random occurrence, but actually trying to investigate how evolutionary pressures cause this process of selection to bring up some of those haplotype sequences. And then we could focus on whether those sequences are associated with a phenotype, right? So for example, if our phenotype is transmission, or a phenotype is kind of manifestation of more acute or more severe disease, those are then the downstream steps that we could try to link the molecular kind of profiles and signatures that we identify with the outcome. But in the context of viral diseases and bacterial diseases, we actually have several different educational projects that we have prepared for this program called Bioinformatics for Infectious Diseases. And so one of the projects included in this program and included in this kind of analysis of viral genomic sequences is this idea of a pathogen that is affected by these haplotypes. So these are some of the other projects that we've covered already extensively in the program, and I'll briefly kind of describe what these are. So there's a project called Origins and Pathogenesis of the Novel Coronavirus. You can find this project on our new portal called learn.omicslogic.com. So for those of you that are new to our sessions, I welcome you to explore that portal, register for free, and explore some of these projects there. We, of course, dealt extensively with Ebola and the pandemic, and the Ebola pandemic in 2014, 2016 is an example of a pandemic where genomic sequencing has been prevalent and used extensively for the first time to characterize almost in real time the spread of the disease and kind of changes that occurred in that virus. And then we have some additional ones that we've used, including an analysis of tuberculosis infection for drug resistance. This is another idea of fitness or low frequency mutations that cause the viral infection to act as a kind of a collection, a swarm of viruses, and their diversity kind of correlating with the fitness, with the ability of the virus to cause disease. And then we also have some other applications in biotechnology for detection of viral strains using next generation sequencing transcriptomic data. So please take a look at these. And then the project that we will be talking about today, as Bipsa mentioned, is about Enterovirus D68. So Enterovirus D68 is a single-stranded positive sense RNA virus that belongs to the Enterovirus genus of the Picornaviridae family. EVD68 infection usually leads to severe respiratory illness and has recently been reported to be linked to the development of the neurogenic disease known as acute flaccid myelitis, or AFM, mostly in children. So what it is essentially the virus going through the respiratory system and entering the nervous system, which is a major concern with polio and other enteroviruses. And so that is a particular type of a virus that can go between these evolutionary and tissue-specific membranes, essentially, that are designed to prevent from viruses to cross over between different tissues. And so this phenomenon suggests that a potential for EVD68 to induce nervous system disease, arousing widespread concern among health authorities and the public, despite the fact that EVD68-associated infection was previously considered infrequent. Since 2005, it has undergone a worldwide burst of growth in the US, Asia, and Europe. EVD68 was first isolated in California in 1962 from respiratory samples of 4 pediatric patients suffering from acute respiratory syndromes. The EVD68 genome is about 7.6 kilobases long, similar to polio, about 8 kilobases long, and consists of a single open reading frame or flanked by untranslated regions. The genome encodes a precursor polyprotein that is further catalytically cleaved, resulting in 4 structural proteins, VP1, VP2, VP3, VP4, and 7 non-structural proteins, 2A, 2B, 2C, 3A, 3B, 3C, and 3D. So there are some drugs that have been approved, and this is based on the understanding of how these bind to the surface proteins that are found where infection happens. So as you can see right here, we have the phylogenetic tree, and so we have the enteroviruses. So the enteroviruses include enteroviruses like EVD, and you can see 68. There are polioviruses, which are enteroviruses C, Coxsackie, so EVB3. And then they're also together with rhinoviruses. So the difference between rhinoviruses and these enteroviruses is that rhinoviruses primarily cause respiratory disease, and enteroviruses typically cause disease in the gut. And that's where the word entero comes from. And so recently they have been grouped together because they actually have a very high similarity in their sequence. So to understand the spread of this recent potential spread of this dangerous genome, let's go ahead and take a look at this next strain link. So I'm just going to go over to next strain. I hope that many of you are familiar with next strain. This is the link. I'll just post it in the chat box. So please let me know once you reach this link. It will show us the evolution of enterovirus from 1985 or 1986, actually, when it was first sequenced in California, as mentioned, and all the way up until now. So please put something like number 1 in the chat box so I know that you are there. Okay, and as everybody is getting there, please let me know if you're already familiar with next strain, put number 2. If you're not familiar with next strain, then don't put anything. So I will know how many people are actually familiar with next strain. Okay, so not a lot of people, I don't see a lot of answers to number 2. So I don't know how many are actually familiar with next strain. What next strain shows is the phylodynamics of enterovirus. And one of the things to make it really dynamic we can do here is we can actually play this sequence to see what happens year by year. So you can see here the slider is moving, and it's showing us the occurrence in United States where it was first detected. Then we see in Canada, in France, we see an increase in the United States, but also on this side, you can see how the clades of this phylogenetic tree start separating into distinct groups as it spreads in Europe, and then you can see in Asia, and then you can see this large emergence of this red circle right here, which is in the United States. And so what we are looking at, these sequences right here, or this particular region, you can see it happened right around 2014, 2015. Okay, so what this shows us is the divergence of specific sequences. What this shows us are the number of those individual genome sequences present by location. So for example, red dots are in the US, and let's say green dots right here are in Sweden. And so we're going to be actually looking at some of those sequences today. Down here, you can see the genome. So remember, we talked about VP4, VP2, VP3, and VP1. So these are the structural proteins, and then these are the non-structural proteins, 2A, 2B, 2C, 3A, 3B, 3C, and 3D. So we just talked about these. what you can see here is diversity. So how specific positions on the sequence have either higher mutability, right? They tend to mutate more or less. And actually, if you scroll down here, you can also download the data. So all this data is available. And for those of you that are interested, you can actually install this to run this on your custom sequences. So this is the phylogenesis of Enterovirus D68. So you can kind of see how it evolved over the years. Go back to my presentation. Okay, so the EVD68 wasn't always in the spot, in the highlight. Poliovirus, the causative agent of poliomyelitis, was discovered in 1908, and poliovirus infection can result in a variety of symptoms, including AFP or AFM, which is acute flaccid paralysis or myelitis that cause lifelong disability and may cause death, right? So everybody kind of remembers polio. And one of the major things that happened were the vaccines against polio. So the first poliovirus vaccine was an inactivated injectable vaccine developed by Jonas Salk in 1955. The second vaccine administered orally was a vaccine developed by Albert Sabin in 1961. Introduction of poliovirus vaccine reduced infections dramatically, with only small outbreaks occurring sporadically. Acute flaccid myelitis, AFM, is a polio-like illness defined by the acute onset of flaccid paralysis in the setting spinal MRA, demonstrating a longitudinal lesion in the gray matter of the cord. So you can see right here, this lesion right here is what is caused by this disease. So that's the clinical manifestation. And the majority of patients with EVD68 infection show symptoms of mild respiratory illness. However, co-occurrences of EVD68 and polio-like neurological symptoms established EVD68 as an emerging pathogen. Before 2014, the EVD68 outbreaks were sporadic and only resulted in 29 cases in the US and 699 cases in Europe between 1970 and 2005. In 2014, the USA experienced the largest known EVD68 outbreak in history, with 120 cases of AFM, particularly in young children. Confirmation of AFM required this MRI scan, and so you could then determine by looking at a picture like this that there was actually a damage in this spinal cord tissue. And so we also know that, as we saw in the previous figure, we saw that the clades emerge. And so these clades have names. And the subclade B1 that is associated with AFM has 12 substitutions that carry the same residues observed at the same positions in paralysis, causing Enterovirus D71 or poliovirus. Subclade B3 is now associated with AFM. The hypothesis states that EVD68 acquired pathogenicity after the Furman strain was isolated. In 2014, there were outbreaks in Europe as well, in France, Norway, and UK. In 2016, a new upsurge in the number of EVD68 cases occurred first in the Netherlands. From many patients, one was presented with AFM. Phylogenetic analysis revealed the isolate belonged to a different subclass than 2014 outbreaks isolates. Okay, so let's take a look again at this next strain right here. And so you will see these names, right? Again, subclade. So you have here B1, B3, B2, B, etc. So what is the history of these clades? So you have right around 2000, maybe 1998, these clades separated into clades A, B, or A and C, so right here, and then this clade separated into B and C. And we can see here how this has a lot of cases in 2014, and now we're talking about a new set of emerging cases in 2016, right around here, And you can see that a lot of them are in Sweden. So these are cases that primarily happen in Europe, not as much in the US. And here you see a lot of these are in the US. Okay, so now we're looking at these different clades. Let's understand what these mutations have the potential to do. So why are we looking for these mutations? Okay, so to understand these mutations, we need to understand the mechanism of infection. So the existence of sialic acid on the surface of human respiratory tract endothelial cells may be the reason for effective replication of EBD68. Neuroaminidase was shown to catalyze the hydrolysis of sialic acid, reducing attachment of EBD68 cells to HeLa cells up to 90%, indicating that it might be a receptor for EBD68. However, some strains of EBD68 from 2014 were resistant to neuroaminidase treatment, and that suggests that there's an alternative receptor. A novel receptor for EBD68 was identified in 2016 as an intracellular adhesion molecule 5, or ICAM5. Okay, so you can see ICAMs are these receptors on airway epithelium, and they also are receptors for human rhinovirus. And here you can see that, again, remember, enteroviruses, the word entero comes from, they enter the gut, and rhinoviruses typically infect the respiratory system. And so here we can see this. So ICAM5 is highly expressed in mammalian telencephalon of the central nervous system, whereas other ICAMs are abundant in lymphocytes, ICAM1 and ICAM3, endothelial cells, ICAM1 and ICAM2, epithelial cells, ICAM1, and erythrocytes, ICAM4. And so that's where we kind of see this change from maybe the regular way that this virus infects humans. And eventually this, you can see here, was published in Cell, Host, and Microbe, that ICAM5 telencephalin is a functional entry receptor for Enterovirus D68. And so because of this growth in cases, it was interesting to investigate how exactly does this infection happen. But after the infection, what happens is that there are virus-host interaction. So when the host cells face pathogenic microbes, different pattern recognition receptors, or PRRs, are expressed by the host cell to recognize viral components through pathogen-associated molecular patterns, PAMPs, that trigger intracellular signaling cascades to activate pro-inflammatory responses. So primarily, we're talking about interferon secretion, stimulating IFN-stimulated genes, ISGs, to activate the antiviral state of the host cell. Enteroviruses are mainly recognized by 3 PRRs, toll-like receptors, TLRs, retinoic acid-inducible gene 1-like receptors, and NOD-like receptors, NLRs. A TLR3 TRIF complex activates a signaling pathway and interacts with IKK-related kinase complexes, resulting in phosphorylation of IFN regulatory factor 3. So once the virus gets into the cell, you have this interferon response that triggers this cascade of immune responses. And what's important to understand is that the non-structural viral proteins like 2A, C, 3A, and 3C can suppress interferon production, allowing for replication advantage. Because what happens in the host cell when the virus enters and it is successfully recognized within the cell, there's this trigger for the interferon response, which causes the cell to shut down production and signal on the membrane to other cells around it that something bad is going on. And so for the virus to effectively replicate, it has to sabotage this process before it can enter its RNA into the production machinery to produce its own proteins, so that the virions can actually reproduce and get out of this cell. And so these non-structural proteins suppress this interferon production. And there are a lot of specifics here. So 3C was shown to cleave TRIF at its amino and carboxyl terminal domains, leading to TRIF's inability to activate NFKBs and interferon gamma signaling, helping virus to escape immune response of the host. Okay, so what comes out of this is that specific cell cycle phases may actually affect viral production. So the cell goes into different phases, and so G0, G1 phase promotes viral production, whereas G2, M phase inhibits it, and it is controlled by EBD68 through regulation of expression of cyclines and CDKs. So cyclin D, CDK2, CDK4, CDK6, and cyclin E are downregulated after viral infection in order to inhibit transition from the G0, G1 phase to S phase, which is a less favorable phase for viral production. And cyclin B1 and CDK1 promote transition from G2M to G0, G1 phases. So as a result, we have this complex regulatory machinery that this virus has, first of all, to adapt to different types of receptors, again, to cause disease in the neural tissue. It has to attach to these specific ICAM5s as opposed to ICAM1s or ICAM4s. And it has to also successfully inhibit this interferon response from the cell recognition to be able to replicate. So this effective replication in the neural tissue in the spine is what causes damage to the spine, but it also causes this kind of evolution from the viral perspective, causing a diversity, right? And so this diversity can be characterized, just like I showed in the first part of this presentation with AMA1, we can think of haplotypes in two different ways. So the first way is when we have consensus sequences from a population, and we are looking at population genetic level diversity. So we're actually trying to combine all of the different variations that we have at the consensus level into a smaller number of groups. So what we did with AMA1 is we had, let's say, 350 sequences, and we reduced that number to about a half, so maybe 150. In this case, when we're talking about viral haplotypes, we're actually talking about an increase in diversity. So for each sample, we actually have multiple substrains, and these substrains are actually called haplotypes because each infection is represented by a group of somewhat diverse viruses. And so the goal here is to look at individual reads and then combine those reads into haplotypes that represent a single sample. Okay, why does this happen? Why does this virus evolution take place, and how? Recombination and mutations caused by the error-prone polymerase that lacks a proofreading mechanism have been recognized as the main mechanism for enterovirus evolution. Recombination plays a crucial role in viral evolution and adaptation by repairing deleterious mutations in genomes, thus rescuing fit viral genomes from low fitness parents. Moreover, it provides the ability for large evolutionary transitions, otherwise known as jumps in the fitness landscape, by producing in a single step significantly divergent genomes better adapted to new environments. The role of recombination in enteroviruses has been summarized in the early 2000s. But since then, a great number of studies have been published providing new insights into the evolution of the members of this genera. And the primary driver behind these new discoveries is this next generation sequencing approach that allows us to accurately detect individual genomes and categorize them as a single haplotype. And the projects that I mentioned before, including the project about polio with low frequency mutations and their ability to affect the fitness of viral populations causing disease, is a good way to get started with that project. Now, if you want to learn a lot more about the biology and the background of this project, we recommend going to this project. It's only $10 on the EDU TBioInfo platform. And for those of you that are already a part of our Bioinformatics for Infectious Disease program, this is included in the program itself. What you will find here is a very detailed overview of everything that I've briefly described today. So let me show you how to get there. Okay, so this is, again, the project. Here you will find the project overview, and then you will be able to go into the background of Enterovirus D68, how it causes disease, what are the infection mechanisms, test your own knowledge about this process, and then get an introduction to the different analysis methods that we will go on to next to discuss how do you actually analyze this type of data. So for those of you that are interested in following along and having all of the background of what I am describing, please go into this project right here. Okay, thank you, Bipsa. Okay, so let's talk about the data. Okay, what is the data that we are going to be dealing with? And before we talk about the data, I wanted to mention that we are going to be using the Tauber Bioinformatics Research Center's platform called T-Bioinfo. And so this T-Bioinfo allows you to process large data sets, including RNA-seq data, perform analysis from a statistical perspective, explore machine learning algorithms that you can apply to different types of data sets, perform multi-omics integration, and provides you with several tools for biological interpretation. So all that we're going to be speaking about today, the practical part is going to be on the T-Bioinfo platform. How do you get access to the T-Bioinfo platform? So the first way is you can register on learn.omicslogic.com. If you don't have an account, just by registering that there for free, Beeps, if you can again post the link, you will receive an email that will give you a demo access to this platform. So this demo access will allow you to try pipelines without actually running any analysis, just to kind of see input, the logic of analysis, and the output. Another way to do this is by actually going to server.tbioinfo, which I will do right now. Okay, and here you can actually request access by filling out this form. Okay, so what I will do again is I will just give here a couple of moments for those of you that do not have an account. So if I may ask that you put into the chat box again, how many of you do not have an account on this platform? Because the whole next section is the section of this workshop is going to deal with a practical analysis of data from this EVD68 infection. So please put in number 3 if you do not have an account on the T-Bioinfo platform. Okay, so it looks like Joseph Christian. So, okay, so Joseph and Kazim Anu, could you please go to learn.omicslogic.com and register there? It will literally take a minute, and you should get an email back that will give you the details of your account. So I'll just demonstrate how to do that for those of you that are just joining for the first time. You go to Learn. Again, I'm actually going to log out real quick. And then what you can do is you can go to register for free right here and create an account. So you can either sign in if you have an account or create an account. And when you do that, you can create a new account. I'm just going to be John, let's see, Smith. And then here, I'll just, use a strong password, and then I can sign up. I'm not going to do that because I already have an account. But once you do that, you will actually receive an email, and it will give you access here, but it will also give you access to the server T-Bioinfo platform. Okay, so I'm just going to give it a couple of minutes, please, Joseph, Kazim, Anu, and Elham, if you could please complete the registration and let me know that you are there. maybe you can just put it in chat, and that way I will be able to see that you're already registered and ready to follow along. Okay, please let me know. Maybe you can put, let's say, number 4 in the chat box. So we're making progress. We already went from 1, 2, 3, and 4. So let me know. I will just give it 1 more minute for you to finish. All right, thank you. All right, so as we are moving along, let me kind of introduce this type of data to you so that you know where this data is coming from. So all of you are probably familiar with, okay, thank you, Kazim. So all of you are probably familiar with Ncbi. So let's try to find this data on Ncbi, and then I will give you actually data that you can use for this analysis today. Okay, so Ncbi contains next generation sequencing and first generation sequencing data from a variety of different types of data. So here I have the link to the bio project. I'm going to share it here on chat. And you can see in this bio project that these are full genome sequencing of European EVD68 samples from 2014, 2016, and 2018, and ongoing epidemics in order to better understand EVD68 evolution and phylodynamics, right? So remember, what we saw on next strain is exactly what we're seeing here. And then you have here 113 Sra experiments. So if you click on this link, you will see here a list of those samples. So let me ask you, for those of you that are following along, what are these data sets? So what do you think was actually sequenced in these data sets? Okay, anyone is just free to put your answer in the chat. We'll kind of see how familiar you are with the different types of data that is hosted here on Ncbi. Okay, go ahead, Sazad.
Sir, the basis of the sequence, I mean the DNA sequence, the basis.
Okay, all right, great. And then Abhimanyu is saying RNA-seq. So blood samples for RNA, Io is saying. Okay, so let's talk about Azad's answer. First of all, he mentioned that this is a DNA sequences of blood samples. So this is a patient sample, but what is extracted from that patient sample is the Enterovirus D68, which, as you remember, is an RNA virus, but it's not RNA-seq in the sense that it measures expression of genes. This is, if we click here, we'll actually see what this data is. Okay, so the DNA preparations were quantified using QuantiTM, dsDNA high sensitivity assay kit, and Tecan Spark 10 M plate reader. They took, let's just get to the interesting part of it. Okay, so here, the library was quantified using QuantiTM, dsDNA high sequencing assay kit, and Tecan Spark 10 M plate reader, and pair-end sequenced. Okay, so it's important to remember, pair-end sequenced to a depth of 100,000, 1 million reads per sample on a high-seq 2500 Illumina sequencer. Base calling and demultiplexing was done using BCL2 FastQ v version 1.87 without allowing any mismatch in the index sequence. So let's deconvolute what this actually means. What that means is that they extracted the viral, so they amplified specifically viral sequences, right? So you can create a primer, amplify that using PCR, and then what you get is an amplified sequence of viruses that are present in a given blood sample. After that, you can see that the sequencing was done to a depth of 100,000. That's pretty good depth. So it allows good coverage across the genome for us to be able to perform this kind of analysis. on high-seq 2500, which is a good length of read and high throughput output from this sequencer. And then, as you know, once you sequence your samples on the Illumina machine, you actually have to combine them because they are combined together into a single plate. So you need to use the barcodes to separate individual samples into FASTQ files. And so from this run, which is the BCL, you get multiple FASTQs. And so they use this script called BCL2 FASTQ, which is published by Illumina. So you can get individual FASTQ files. So this is why it is called amplicon, and it is of viral RNA through RT-PCR, and the layout is paired. So why do we use paired reads to be able to analyze these sequences? The reason is because it provides us with more reliable sequences and the ability to perform error correction. Okay, so let's go on. Let's take a look at the specific. So as you can see, there are 113 sequences here. And so out of these sequences, we actually want to select specific ones. Okay, so let's take a look at which ones do we want to take. You can see that we have 1, 2, 3, 4, 5, 6, 7, 8, 9 of them. And you can see that they come from years 2014, 2016, and 2018. So we actually want to take samples that represent this change happening over time. Okay, let's go back really quickly to next strain. Okay, so this is our 2014 right here. Then we have 2016 right here. And then we have 2018 right here, right? So those are the samples that we would like to take, 2014, 2016, 2018. Now, we also have matching samples that are gene bank. So these are assembled consensus sequences. They're not FASTQ files, which contain individual reads. These are actually assembled files that allow us to know the consensus sequence or the most prevalent sequence in a given file. So these are GenBank files, and GenBank files have also the reference genome, which was first sequenced, as you remember, in California in 1962. All right, so to gain access to all of these files, I'm not going to bother you with actually finding all of these individual sequences, but I will just first show you how would you go about it. How would you, if you find a data set on Ncbi, how do you actually download the sequence IDs so that you can process them on the T-Bioinfo platform? Okay, so the way to do this is you go to Ncbi right here, and what you can do is you can send results to Run Selector. The Run Selector provides you a view of all of the different types of metadata and your sample IDs right here. You can see where it was collected, spot length, average length, and stuff like that. And so what you can do is you can download metadata. The metadata will provide additional information, for example, location of data collection, year of data collection, etc. But here it's useful because you can actually navigate between the different pages because we have so many samples. And let's try to find some of these samples real quickly. So as you can see, we have here 5074, 5075, 5076. Okay, let's try to find these. So here we have 5074, 56, 4, 5, 6, 50, 54, 56. Okay, so 50, 54, 56. Now I can go on top over here, and well, let's get the rest of them. So 50, 54, 56, and then 44, 96, and 100. So let's find 44. Again, then 96 and 100 would actually be on the next page. So go to the next, okay, well, let's keep it at this. So we have 1, 2, 3, 4, 5, 6, 7. So we have 7 samples right here, and I can get the metadata to see what is in these samples. So what I get is a file called sratable.txt. If you look inside this txt file, you can see that actually everything is separated by comma. So you need to change it to a CSV, which is what I did here. And now you can see what are the specific things, like, for example, all of these were from Sweden, right? And then you can move over here, and you can see that indeed they were all sequenced by Illumina HiSeq 2500, and these were all Enterovirus D68. And then here you can actually see the strain. So here you see B3 strain, which are the first 4 samples, and B1 strain, the second 3 samples, right? So they're actually different. Let's see if they give us, okay, and they give us the date. So this one is from 2018. These 3 are from 2016, and these 3 are from 2014. Okay, let's go back and again check with what we know about these strains. So here we have B1 strain. Here we have the B3 strain, right? So what we see is that the B1 strain, which was predominantly in the US, just kind of died off, right? There have not been any new samples collected after 2014. But the B3 strain is kind of more fit. It's more able to continue and replicate and cause these outbreaks over the years. And so we see that first, indeed, it happened in 2014 at the same time that we saw this outbreak. Then it moved on, and we see this other outbreak in 2016. And then we have this other outbreak in 2018, which is also predominantly B3. Okay, so what if we wanted to take these files and upload them to the platform? So here, let's forget about all the other information. I only took the IDs of these links, and let's go to the platform. Now, for those of you that are using the demo platform, you actually cannot upload the files yourselves. You can use the demo pipeline, but I will, for those of you that do have access, I'll just demonstrate how this works. So out of this whole section of areas of analysis where we have next generation sequencing, data processing, here we have a section for data integration and modeling, and one of those is on the phylogeny. So let's go to this phylogenetic tree. You can see that we can map our data to the reference genome, and here we will be able to determine the haplotypes. But before we do that, does somebody remember whether these were FASTQ files and whether they were single end or pair end? Does anybody remember what they were? All right, Io is paying attention. Paired end, thank you, Io. So we will change this right here to paired end. And then I have to go to step number 2, which is upload next generation sequencing data. Now, I'm not going to download multiple megabytes and gigabytes to my computer. Instead, I want to directly upload them from Ncbi. So right here, I have Ncbi run table upload, and I can just go to add files and select this accession list right here to upload it to my pipeline builder. So now I have to separate them into groups. Okay, how would you propose to separate them into groups? What are some phenotypic categories that you can think of that could help us separate them into groups? All right, excellent. Rashid is also paying attention. That's group. That's good. Rashid, maybe you could suggest, how would you separate them into what groups? Would it be 2 groups, 3 groups? All right, excellent. Thank you, Io. 3 groups. So let's create 3 groups. Now let's go back to our table, and we'll take a look at what are the samples since he said by year, we'll do it by year. So we have this first sample. This is 5044. So 5044, we'll drag it to 2018, will be our group C. Then we have here 2016, which are 50, 54, and 56. So 50, 54, and 56. And here we have the rest of them, which are all 2014, and they are 74, 75, and 76. So 74, 75, and 76. All right, so now we have outgroup for framing. Okay, what could that mean? What does it mean to have an outgroup for framing? So an outgroup for framing is a group of files that allows us to associate a particular haplotype, a set of co-occurring mutations with a defined consensus genome, right? And so here I'm just going to get my links. For those of you, that are following along, I will give you the links. Okay, there we go. All right, so here are the links for those of you that are interested to kind of perform the full analysis. These are the links to the individual tables that you would need. The first one is the SRA run table. We showed you how to do that. And then you see here we have two outgroups for 1962, right? That's the reference genome, and the 2013 consensus. Okay, so let's go and take this. This is a gene bank file. Okay, so you can see this is a gene bank file. It has the translation of these sequences and nucleotide to amino acid sequences. So let's go ahead and say file, save as. Okay, I'm just gonna save it to downloads. Okay, and here I don't need the txt, so it's a GB. And then the second one was this one. Okay, now I could download them to my computer, right? And then I have to add those files, right? So let me, this is outgroup 2, file, save as. See. And now I need to add them right here. All right. Okay, and then here I can add the other one. And then I need, and then I need the viral genome. So again, I have that right here. Okay, that is the reference genome. So actually, I don't think I need it, but this is the reference genome. All right, now we're going to build our pipeline. So first of all, you can read a little bit more about phylogenetic analysis here that can either work with FASTA files or FASTQ files, essentially looking at multiple sequence alignment and phylogeny based on consensus genome, or what's called subconsensus analysis, which is looking at what's within those individual reads and analyzing that data together. So I'm going to click on OK. And now you can see that each job is organized into different types of sections. Okay, and so here you can see that I have mapping. So I will use Bowtie 2. Bowtie 2 is an algorithm that has been optimized from Bowtie 1 to map efficiently using optimization of memory usage for mapping onto the reference genome. So I'm going to use this one. And then here you can see that I can create sample consensus. Okay, so if I wanted to take all of my reads and create a FASTA file from each one of those next generation sequencing read raw files for FASTQ, I could go to here. But instead, what I will do in this case is I will skip alignment to the reference genome and go directly to haplotypes. And this is a method called Sra, maybe? It is a computational method for inferring the structure of haplotypes from sequencing of mixed samples. So again, if you imagine multiple types of viruses combined together in a single blood sample, how do we now separate them into the actual viral sequences that we expect to be present there? Okay, so here we have some specific settings, including the window size, because again, we're looking for a specific combination of mutations that can categorize essentially an assembled genome. So we're sort of performing an assembly based on all the sequencing reads that we have. Okay, and so here we'll click on save. Then we'll perform multiple sequence alignments. We'll convert that multiple sequence alignment to amino acid sequences. We can also look at specific mutational profiles in the third codon position, which we don't need to do in this case. And then we have a series of phylogenetic methods from multiple sequence alignment using Bayesian or neighbor joining algorithms like APE, which provides us with several different options there. So we will use the Coalescent option. What is the coalescent constant size? It assumes that unknown constant population size back through time. So essentially what we are looking at typically is the divergence, and coalescence meaning their common ancestor sometime in the past. And since coalescence is a process directed backward in time, in opposite direction of the divergence, it is not advised for speciation times estimation. So in other words, typically you would use it for evolutionary relationship, not for divergence over time with a specific time allocation. Okay, so then what are we using here? We're using nucleotide, and we will click on end, and we can save the pipeline. And click save and run this on the cluster. All right, so once our pipeline has started, we will have to wait a little bit for this to be completed. And now let me skip on to the results. Okay, so for those of you that are following along, here's a link to a pipeline that has finished. So this pipeline that has finished provides us with several different outputs. So here you can see the input was translated into a FASTA file. So let's take a look. The FASTA file and the cluster files are almost identical and allow us to view the consensus sequences aligned together for all of the haplotypes. Remember that this is for all of the haplotypes. Then you can see that we have a PDF file of the phylogenetic tree. So let's take a look at that PDF file. Okay, so here we have a PDF file, and Harpreet, did you have the time to prepare something to speak about these results?
Yes, Ilya.
Okay, so I will pass it on to Harpreet to describe the results of this pipeline.
Okay, Ilya, thank you. Can you just make me co-host? Okay, thank you, Ilya. So now let me share my slides. Sorry, this screen. Okay, so I have already downloaded this PDF file. So if I just go to this, so here what you will find, like if this is like a phylogenetic tree. Now, if you look at this phylogenetic tree, you will find three major clades, like this one, and this one. So here, if I show you what it contains, like here, these are the first one, it's showing the outgroup. So this is the outgroup that belongs to 2013. Then we have a reference genome. So then this first outgroup that is belong to the year 1962. So then it is making completely distinct cluster, completely distinct clade. So then this is this outgroup have taken from 2013, and then this is group 1 sample. So if you remember, we have taken samples from 3 different years, like 2016, 2012, and 2014. So here each year is actually making a different clade itself. Here within this, you will find multiple haplotypes. So like here for the group 2, which is 2016, so sorry, 2013, and this is the third group, which is 2016. So we have all the 3 groups from 3 different years. And what we have observed, haplotype of each year actually clustered together on this particularly single phylogenetic tree. And each year, these haplotypes from each year also making a completely distinct clusters. So then this particular outgroup, that is outgroup time point. So this is the different how you can find actually which group is close to which year. For example, this particular reference genome is also taken from the year 2013, and this outgroup is also taken from 2013. So they are more or close to the 2012. So then completely the 2000, this is 2018 and 2016. So we can also understand what is the relationship between these haplotypes as well as these different clusters. So whether they are different from each other or not. So this is based on the phylogenetic analysis itself. So then apart from that, we have here on in our results, if you want to understand what are the mutations are taking place between the samples from different regions. So this is, if you just download this table, that is full table TSV, so which ending here. So you will find also what are the important mutations maybe are in these groups. So one can find out what are important mutations, and then that is at nucleotide as well as amino acid level. So here, like at each position, what are mutations are taking place in each of these haplotypes. So you, if you are further want to analyze this, you can find what are different mutations, like over the years from 2012 outbreak to 2018 outbreak. What are the important mutations that are taking place between these kind of samples? We can actually analyze that at the haplotype level itself. So these are the results that I just want to suggest. If you have any other question, you can ask, and please go ahead and ask if you have any question regarding phylogenetic tree or this mutational analysis.
All right, thank you, Harpreet. And so for the purpose of time, because we don't have a lot of time left, I want to actually pick up here to talk briefly about the significance of this type of analysis. Why is it useful? What is it useful for example, right now? So as Harpreet explained, you can see how the different groups by year are separated into a certain number of haplotypes. And so the immediate outcome here is you can start looking at how divergent each group is. And you can see that this group has a smaller number of haplotypes as opposed to this group in group number 3. So you can just count the number of haplotypes, and that is one method of inference. What do you get out of this type of an analysis? Another one is that these right here are proportions. So these numbers are proportions of how many reads are proportionate to the total number of reads that contain these haplotypes. So you can look at the number of positions that are mutated per each haplotype, because as Harpreet showed, we have this full table of sequences and their coding and non-coding mutations. And you can start comparing how diverse they are and where the specific positions that are influenced by this evolutionary process. Now, this was an example with EVD68, but a similar story is known to happen even with SARS-CoV-2, which we're all familiar with. So interestingly, there was recently a publication that describes quasi-species in the context of SARS-CoV-2. So here you can see the representation of the impact of comorbidity on SARS-CoV-2 quasi-species generation and testing. So here you can see how comorbidities and different types of symptomatic and asymptomatic response could allow a different type of evolutionary pressure on these communities of viruses causing disease, eventually causing the spread as well. And so one of the things that has been proposed is that different populations with different comorbidities, different types of phenotypes, different kind of genetic background could cause diversion between these quasi-species and a lower level of detection by the common standard method of RT-PCR. So it's a very important topic, especially in pandemics like today, to be able to link the diversity, the diversity of phenotypes, not only to a specific clade, but also to the diversity within that clade, which could be defined by haplotypes. And so that's why we're also going to be looking at how to utilize some of these similar types of analyses. You can see here that we did today Sra, right, analysis of haplotype consensus sequences, with a more general approach that will allow us to detect low frequency mutations that could indicate prevalence of specific variants. And so in conclusion of today's webinar, I wanted to let you know that coming up, we have another webinar on the genomic surveillance of SARS-CoV-2 to detect variants of concern, right? So how can we utilize this type of an analysis to actually be able to find and understand the trends behind the emergence of SARS-CoV-2, to perform genomic surveillance, how to collect data, how could it be sequenced, and what are some of the bioinformatics analysis methods that allow us to detect these important variants of concern, how to perform downstream analysis of genomic data with evolutionary studies like we did today, but also to trace origin and to report the variants of concern or specific clades based on different nomenclature that's out there, like pangolin and others, to be able to report about presence of specific clades. And then just to build a report. How do you actually build a report? What are some of the considerations around coverage, specific proportions of variants, positions? How do you identify positions to look for these clades? And then also to discuss some future directions for large-scale vaccination and variant of concern monitoring. So again, as you know, there are many different distinct SARS-CoV-2 clades and variants that define them. These have been over the time separated into more and more numbers of clades, and so it's important to continuously monitor those. Here's an example where you have specific non-synonymous and synonymous mutations that define. This is the 20I or the B117 UK variant. You can see that it started in the UK about November 2020, and it's prevalent today in the UK, but also starting to appear in other areas like the US. So you can see here different clades in the US. And these are the mutations in the spike protein that, of course, potentially have an implication for the different types of infection phenotypes that we're observing in the population. Now, to detect these, we need to understand the genomic organization, the specific mutation rates that are of interest and are of concern, as well as some of the validation of those variants that, for example, here you can see for the amino acid 614 G to D that has been confirmed with higher transmission rates. So we'll actually take a look at the structure and organization of the genome, the particular regions that imply specific function, how this function could be monitored and understood in the context of genetic background of different patients. Then how we can actually link the mutability of individual positions with an understanding of those regions that are highly variable, where we expect these variants of concern to be to get established, and what is the effect of some of the selective pressures as we have the large-scale vaccination and treatment of SARS-CoV-2 infection with symptomatic and asymptomatic cases, as well as different types of variation that just emerge as a result of this long-term infection that is observed. Now, importantly, it has been shown that this has an impact on the ability of vaccines to cause a long-lasting immune response. And so this is not just to understand what's happening, this is also to be able to anticipate how these changes will impact the future rollout of vaccines and the modification of these vaccines with potential boosting shots that we'll be expecting. And so we'll talk about some of these variants of concern. We'll also talk about some of the additional things that are not as highly described in literature, not just mutations, but actual long deletions that are observed in long-term kind of infection studies. And we'll talk about the National Open Genomics Consortium for the COVID-19 Response, or SPHERES. What is NIH? What is CDC doing to address some of these topics? And so if you want to learn about this consensus level analysis of variants, detection of novel prevalent variants, subconsensus and low frequency variants, detection of variants of concern in low quality samples, for example, in wastewater, and tracking the spread and the emergence of variants in densely populated areas, we invite you to join this upcoming webinar and actually a program, a whole program. And thank you, Bipsa, for posting this link in the chat box. This is the link for you to learn more about this and to register for the webinar. You just have to submit your information right here. And then we will again email you some of the details about how to actually perform this analysis. Now, importantly, we're not just going to be talking about it. It's not going to be, so the webinar is going to describe the topics, but we will be doing some hands-on analysis, starting from describing different methods of sequencing, looking at files and understanding what types of files they are, looking at different ways we can analyze this data, including specific types of funding opportunities that you could pursue if you are conducting such type of sequencing and analysis. Examples from the studies that we've been involved in for wastewater sequencing, methods of analysis on the T-Bioinfo platform for an in-depth characterization of variants and what they do, how do they impact, and how to measure the impact using different types of metrics. And then we'll also look at actually preparing a report on your own using the outputs of these pipelines, but only focusing on particular important features that include analysis of per protein sequence analysis, different types of sample analysis, and then also the impact of pre-processing, as you can see right here with Trimmomatic and PCR Clean, and some reporting, how to write a script in R so that you can highlight the important features. And this is going to be based on a sample report that we'll download from Cosmos ID, which is a company that offers to generate reports for SARS-CoV-2 clinical samples. So we'll make a report exactly like what they do based on coverage and specific variants of concern, like you can see here, the UK variant. And so we'll do that in R using ggplot and using some additional libraries that are going to be useful for data manipulation. So thank you again for joining in today. We look forward to seeing you in this upcoming webinar that's going to happen at a future date. And I'll pass it on to Bipsa to talk about the details. Thank you.
Thank you so much, Ilya, for that overview. So let me go ahead and share my screen again so that I can introduce you to the upcoming webinar. So as you know, we are going to be doing hands-on sessions. So if you have not yet created an account, please go ahead and create an account on learn.omicsLogic. That will also give you an access to the TBioInfo platform. And if you're facing any problem, you can reach out to us at marketing@pine.bio for setting up that account, so that by our next webinar, which is going to be happening on April 29th. So the webinar is going to be on April 29th, and these are the dates of the program. So let us just take a quick look at the different dates of the program. So we will be starting on 3rd May, which is Monday, and going on till Friday. And these are the topics that will be covered in this workshop. And the total cost of this workshop is only $50. So if you're interested in joining, again, you can email us and we will be sending you a link to join this workshop. But for the webinar next week, you just have to fill out the form and we will be sending you details of how to join both the webinar as well as the full workshop. So besides this workshop on SARS-CoV-2, we also have some programs coming up, which if you are interested, you can join. So we have the Biomedical Data Science and Drug Discovery program that is starting on April 26th. This is a full 4 months program, which is going to be starting on April 26th with genomics, then with transcriptomics on May 19th, June 14th, we will start to talk about data science, and from June 12th to July 19th, we'll also have sessions on chemoinformatics. Again, if you need more details regarding this, you can fill out the form at the end of the page. And the third program that we have coming starting this Friday is a 2-day workshop on precision oncology with a focus on lymphoma data. This we are doing in collaboration with the All India Institute of Medical Sciences, New Delhi. And the cost of this workshop is 2,000 Indian rupees. So if you're again interested, you can register for this webinar and register for this workshop. And you can reach out to me for any details. For all of you who are interested in being updated about all of the upcoming free webinars as well as sessions, we have this page which you may have already registered in. So we'll send you the updates to join these sessions. And again, if you have any questions, please feel free to reach out to us and post them in the chat box, especially related to whatever we have spoken about today. And when you run those analysis on your own or explore the platforms, if you come across any questions, please feel free to post those questions to us at marketing@pine.bio. And I hope that you have followed us on our Facebook and LinkedIn pages. We are active on both these platforms as also on Instagram, and you will be able to not only learn about the upcoming programs and sessions, but you'll also be able to work and interact with a lot more people who come from bioinformatics, biotechnology, and life sciences domain, and gives you an opportunity to learn about all the interesting research that is going on around the world in related to bioinformatics. We keep on posting about interesting articles, publications, and topics in these groups. So please keep a watch out there. So I hope that you really enjoyed today's session. We hope to see you at the upcoming webinar on the variant analysis and detection of SARS-CoV-2. And thank you all again for joining today's session. Have a wonderful day ahead. Okay, so the cost for the AIMS workshop is 2,000 INR. So if you are not in India, you can convert it to the local currency to see what is the cost. But as this is organized in collaboration with us with the AIMS New at Delhi. So it is priced in INR. If you have any questions or you want to join the workshop, we still have one more day tomorrow. So please reach out to us. And okay, so the total cost would be $26.53 US for this workshop. It's only a 2-day workshop, and it will be from 10 a.m. IST to 5 p.m. IST, which may be from the Western Hemisphere. It may be night time for you, but we will of course be recording the sessions and sending it to everybody. So if you're still interested in joining, please let us know, and you can reach out to me for any of these details. So thank you all again for joining. We'll send you the recording of today's session as well, and you can find it posted on all of our social media very shortly. And we'll see you at the upcoming sessions. Have a lovely day.